Hello, everyone. I'm Dr. Aparna B Gunjal. I'll just say that in short, my introduction. I'm working as the assistant professor in the Department of Microbiology, Dr. D.Y. Patil, Arts, Commerce and Science College, Pimpri, Pune, Maharashtra, and I'm from India. The title of my talk is Antioxidant and Bioautography of Medicinal Plants, Eugenia jambolana and Asparagus racemosus. So this is the title of my talk that is there. If you see that I hope my screen is visible. Yes, I just want to confirm.
Your screen is perfectly visible and we can hear you.
Okay. So plants, we know that plants such as the vegetable, fruits, spices, medicinal herbs, so they have been used to cure many diseases since the ancient time. The diversity of secondary metabolites isolated from plants have shown the presence of phytochemical compounds. And these phytochemical compounds, they have anti-cancer, antibacterial, analgesic, anti-inflammatory, anti-tumor, antiviral, as well as many other activities. During the past few years, there has been a dramatic increase in the microbial resistance to the antimicrobial agents, even due to the use of the antibiotics, because antibiotics, due to the repeated use of the antibiotics, now there is antibiotic resistance. Plants are among the most common and important sources of valuable compounds, such as the phytochemical compounds and even some of the drugs. Therefore, there is an urgent need to study the biological properties of medicinal plants, various medicinal plants, so that they can be used to develop new drugs so as to help in the cure of diseases. If we see the medicinal value of plants, it lies in the bioactive substances, which we can call it as the phytochemical compounds. So which produce definite physiological action on the human body. The different parts used include, because various plant parts we can use. So it includes the roots, the stem, the flowers, the fruits, the twigs, and other organs that are there. The phytochemical compounds, they include, because there are categories in it, different categories. If you see, they include the flavonoids, the phenol, the phenolic compounds, the glycosides, the saponoids, the alkaloids, the tannins, the amines, terpenoids, beta-alanines, and various other metabolites that are there. So that is why the use of the plants, due to the presence of these various phytochemical compounds, is gaining the attention for the development of the new drugs that is there, so that it can be used to cure many of the diseases. And now research is gaining importance in this field. Plants, they contain phytochemical compounds which supplement the need of the human body by acting as antioxidants. So some of them, they can act as the antioxidants also, if you see. The consumption of fruits and vegetables has been linked with several health benefits, a result of the medicinal properties and high nutritional value. Besides the phytochemical compounds, even some of the proteins, the vitamins are there present in the plants, so which are giving it wide nutritional value. Exploitation of the cheaper sources of antioxidants, especially from the plants, has gained much importance. Antioxidants present in the plant extracts as phytochemicals, they act as the free radicals, which will help in scavenging and neutralize them, protecting the cells from damaging effect of the reactive oxygen species. A bioautography assay has helped for the detection of antimicrobial and other natural compounds. So it facilitates the directed isolation of various active constituents, that is the primary as well as the secondary metabolites, which are present in the plants, as well as it also enables the localization of bioactive compounds on a chromatographic plate where you can apply the microorganism. This is a table, if you see, which mentions about the medicinal plants and the uses. The first is the scientific name of the various plants. The second column, it represents the common name. The third is the part of the plant used and the various uses. The scientific name, I will just read the scientific name: Garcinia pedonica, Ocimum basilicum, Phylocanthus thyriformis, Ziziphus mauritania, Eugenia jambolana, Androphes paniculata, Terminalia arjuna, Asparagus racemosus, Chlorophytum borevillianum, and Vittenia somnifera. So if you see the common name also that is given, and various parts of the plants, they can be used. It includes the fruit, the leaves, the seeds, the inflorescence, or sometimes even the entire plant can be used. The bark, the tubers or the tuberous roots, the rhizome that is there, even the bark or fruits, the inflorescence also, they can be used. And the uses, because these include the phytochemical compounds, so they can be used to cure various diseases, as well as they can be used to cure the respiratory disorder or the fever that is there, or the bleeding disorders, or the diarrhea that is there, or the cough, the weakness, or even the ear pain that is there, the digestive properties. And some of these are the uses, which is why these medicinal plants are becoming important. If you see the work that I'm representing here, the objectives included work: selection of the plants, basically Eugenia jambolana and Asparagus racemosus. So what was done that these plants, they were collected basically from the India, that is they were collected from the UP, Uttar Pradesh side, and extract of these plants, they were prepared in different solvents that is there, such as the hot water, the cold water, the chloroform, isoamyl alcohol, benzene, N-hexane, petroleum ether, and the diethyl ether. Because we require to prepare this plant extracts. Once these plant extracts are prepared, then the bioautography and the antioxidant activity of this was done. So the collection of the plant material, so this was taken from the nursery, the seeds and the tubers of the plants, they were taken of Eugenia jambolana and Asparagus racemosus. They were washed under the running tap water and then air dried so as to remove the moisture. The seed of Eugenia jambolana and the tuber extracts of Asparagus racemosus were taken. After these were collected, to prepare the plant extracts, they were first dried and then they were made into fine powder using the mortar and the pestle as well as the mixer. So plant extraction was done using the cold water extraction, the hot water extraction, and the various organic solvents such as the chloroform, benzene, isoamyl alcohol, petroleum ether, diethyl ether, and N-hexane. The antioxidant property after the preparation of the plant extracts, the antioxidant property of this extract was studied, because this antioxidant property, it helps to scavenge the radicals that is there and thus protect the cells from damage. So the plant, for this antioxidant property to study, the method used was ferric reducing antioxidant method. So the plant extracts in different concentrations such as 10, 25, 50, 100 milligram and 1 ml of the ethanol was taken, and 0.5 ml of the phosphate buffer was taken with pH 6.6. To it, 2.5 ml potassium ferricyanide of 10 gram per liter was added and incubated at 50 degrees Celsius for 20 minutes. After that, it was extracted using 1.5 ml TCA, that is trichloroacetic acid, at a concentration of 100 grams per liter. The mixture, the contents, they were properly mixed, centrifuged at 3000 RPM for 10 minutes. After the centrifugation, the supernatant that was obtained, 0.5 ml of it was taken, to which 1 ml of the distilled water was added and 0.5 ml of the ferric chloride was added, because it is a ferric reducing antioxidant method. The absorbance was read at 665 nanometers. Increase in the absorbance of the reaction mixture directly indicated the antioxidant activity or the reducing power of the plant extracts. After the antioxidant activity, the bioautography was also studied by immersion or agar overlay method. For bioautography, the plant extracts, they were spotted on thin layer chromatography plates, which were taken from the Merck company that was available. Solvent system for the thin layer chromatography used was chloroform is to ethyl acetate is to formic acid in the ratio 10:8:2. After the spot inoculation, the TLC plates, they were dried under stream of air for the removal of residual solvents. And inoculum was prepared in the nutrient broth. After the inoculum was prepared, it was also standardized. The cell count was standardized using the McFarlane turbidity standard method. TLC plates, then they were placed directly in the sterile Petri plates and then covered with 4.5 ml of the inoculum. Inoculum was previously cultivated. Plants were incubated, the plates were incubated at 37 degrees Celsius for about 15 hours and then sprayed with aqueous solution of phenol tetrazolium chloride at 2 milligram per ml. The plates were then re-incubated at 37 degrees Celsius for 1 hour. Clear zones on chromatogram indicated the growth inhibition. Now the antioxidant activity, because it was studied, so the antioxidant activity of the plant extracts, Eugenia jambolana and Asparagus racemosus, was seen, and it was compared with the standard, that is the hydrogen peroxide. So there was increase in the absorbance, which was seen at 665 nanometer when it was compared with the standard also. So the increase in the absorbance showed the antioxidant activity. A bioautography, the test organisms that were used were E. coli, S. aureus, Serratia marcescens, Klebsiella pneumoniae, Proteus vulgaris, Pseudomonas aeruginosa, and Bacillus cereus. So the RF values that were obtained were mentioned here, which were 0.56 for the E. coli, Klebsiella pneumoniae 0.54. In bioautography procedure, the selected plant extract, Eugenia jambolana, prepared in chloroform, so there was spotted on thin layer chromatography. And after the agar-lay procedure, the reagent used was phenyl tetrazolium chloride. It showed the development of dark red color spots. And after 1 hour incubation, the zone of inhibition was obtained on the thin layer chromatography plate against the dark red background. So we can, the conclusion, the interpretation was the antioxidant activity of the crude extract of plants, it depends on the presence of phytochemical, because we also studied the presence of phytochemical compounds also. And the bioautography, which was done, it helped to understand the presence of antimicrobial compounds, which facilitated the further isolation of active constituents which were present. Thank you, and I thank all the viewers, I can say, and the team of Magnus Group